So in chapter 8.3 of your textbook, we are introduced to the concept of the regulation of cell division. So in the cell cycle, we know that there are three phases. We have G1, synthesis, and G2 making up the interphase, then mitosis being the second phase, and then cytokinesis being the third phase. Now, the entire cell cycle is regulated, which means that it is looked after and it is controlled. So we're going to look at exactly how it is regulated. So at VCE level, you need to know that there are three checkpoints. So three checkpoints within the cell cycle. All right. So if we look at the cell cycle, so we have here, for the most part, we have interphase, which is made up of G1, S and G2. This is the S phase here. G1, G2. We have mitosis here. And cytokinesis. So if we have our little cell cycle here, now it's not drawn like entirely to scale, so G1, S and G2 aren't like completely proportionate, but we can find three checkpoints. We find one checkpoint at the end of G1. We call this the G1 checkpoint here. This is the first of the checkpoints. So the G1 checkpoint is actually one of the most important checkpoints. If a cell passes the G1 checkpoint, so it passes it meaning that everything checks out at G1, the cell becomes fully committed to cell division at that stage. So if a cell passes this checkpoint, it becomes fully committed to cell division. All right. And if it fails, it might be arrested into G0 phase, which is something else. But okay, so this is the first of the checkpoints. So what happens during the G1 checkpoint? Well, G1, because it occurs at the end of gap 1, and gap 1, the role of gap 1 is to prepare a cell for the replication of DNA and also generally just cell division. G1, you see the growth of the cell, the growing of, the duplication of organelles, resources being gathered, and so on. The G1 checkpoint is just to make sure that all of those things have occurred as required, especially the gathering of resources for DNA replication. So DNA replication resources are checked. So what do we mean by DNA replication resources? Well, this can include the raw material for DNA, which are nucleotides. Do we have enough nucleotides? Do we have proteins that are involved in DNA replication? Do we have enough of those? And DNA replication also uses a lot of energy, so the cell needs to make sure that there's enough energy reserves in order to carry out the replication process. So in G1, at the G1 checkpoint, all of these things are checked. And if there's not enough of any of those, the cell will continue, or it will go back a little bit and it will start to solve the issue. So it will pick up more nucleotides if it doesn't have enough nucleotides, or gets more energy if there's not enough energy. So that's the G1 checkpoint. Making sure all is good before DNA replication begins, because the S phase is all about DNA replication, isn't it? The synthesis phase, DNA synthesis. Okay, so that's the first of the checkpoints. This is checkpoint number 1. The second checkpoint occurs someplace in the G2 phase. So this is called the G2 checkpoint. Now the G2 checkpoint is somewhat similar to the G1 checkpoint in that the DNA is checked and also the proteins, certain proteins are checked. But the focus is different. So since DNA replication has just occurred, we're not going to be needing proteins anymore for DNA replication. Instead, now we have to look at proteins that are involved in mitosis. Do we have those proteins? Do we have enough of those proteins? Do we have the energy in order to carry out mitosis? What other things are involved in mitosis? We have the mitotic spindle, the centrioles, the centrosomes. Do we have all of those? So in the G2 checkpoint, the resources for mitosis are checked. So the resources for mitosis are checked. So that could be proteins involved in mitosis, energy, because a lot of energy is required for mitosis. But one of the most important things that is checked in G2, I'm going to give this a star because it's really important, is that there are no errors in the DNA. So during the synthesis phase, the DNA was replicated, and it's absolutely essential that the DNA is perfectly replicated. So the DNA is checked at this stage, at this checkpoint, for perfect replication. If there is DNA damage or like irregular replication that has occurred, at this stage, at this checkpoint here, that needs to be fixed. So the cell does have mechanisms for repairing imperfect replication of DNA. And that is mostly done at this point. So this is one of really the most important functions of the G2 checkpoint. Now, if this DNA isn't checked and it continues to go through cell division without like perfect replication of that DNA, what you essentially get are mutations in your DNA. So every cell that is a daughter cell going forward, so every descendant cell from this cell here that would have an imperfect DNA replication step is going to carry like errors in the DNA. And those errors can accumulate. So later on we'll look at what happens when those errors do accumulate. All right, so that's the G2 checkpoint. And the last of the checkpoints we find during mitosis, specifically at metaphase. So we call this the M checkpoint. M for mitosis or M for metaphase. But the M checkpoint is the third one. So what happens during the M checkpoint? Well, in order to know what happens at the M checkpoint, we should consider what happens during metaphase, because we know that this checkpoint here occurs at metaphase. At metaphase, we have the chromosomes all lining up at the equator of the cell. And the M checkpoint is just to make sure that all the chromosomes are lined up properly. So chromosomes are checked for alignment. Checked for alignment. So if we have our cell here, we're going to make sure that all the chromosomes are properly aligned like so. So if you have like one chromosome that is not lined up like on its side or something like that, that needs to be corrected. So the cell would stop mitosis for a moment just to make sure that that is fixed. Another thing that is checked is that the spindle fibers are correctly attached. And this is important This is important because the spindle fibers are key in like the separating of the chromosomes, the chromatids. So if you don't have those spindle fibers correctly attached, you might not have proper separation of the chromatids. And what's the result? We have non-disjunction as like the consequence of spindle fibers being incorrectly attached. So this is a key step. All right. Whoops. So these are the 3 checkpoints absolutely necessary for there to be minimal damage that accumulates as a result of cell replication. So these are really important. So I would tabulate these 3 different checkpoints and what occurs in each of the checkpoints. And really this is just like scratching the surface of what happens, of what is really covered in your textbook. So making sure you're looking through your textbooks just to like get all the extra information. Okay. So just having a look at the learning goals, we have talked about generally why checkpoints are needed. They're needed because we need to make sure that cell replication occurs with the minimum amount of damage being carried through. We've identified where they occur and we've outlined each of their roles, but we haven't yet described what happens when a cell repeatedly fails at a checkpoint. Now, when a cell goes through a checkpoint or stops at a checkpoint and it fails a checkpoint, the cell will try to fix the issue. But sometimes the issue cannot be fixed even after multiple attempts. Or it gets to the checkpoint, the cell checks it the first time and it goes, I don't think we'll be able to fix this problem. So what happens when a cell fails at a checkpoint numerous times or has too much damage to begin with? We have this special process called apoptosis. Now, apoptosis is programmed cell death. Programmed meaning like a very specific, very systematic process by which a cell dies. So programmed cell death is the result of checkpoint failure. So if you want to have a look at exactly what happens in apoptosis, your textbook covers it in some detail. But okay.